So here we have a cell in interphase, and just by looking at it, I know it's an interphase because the way the DNA looks. So the DNA is uncondensed in this form, and it's in the form of chromatin, which is DNA and proteins. And what we're going to see is we're going to see our cell go through G1, S, and G2, which all together make up interphase. So, one thing, during G1, our cell grows. During S phase, the DNA duplicates or doubles. And in G2, it gets ready for division. So let's go ahead and see what happens as we go through each of those phases. So here's G1. You can see the cell membrane. The cell is increasing in size. And in S phase, the DNA doubled. So let's watch that again. G1, the cell grows. S phase, we double the amount of DNA. And then in G2, we're preparing to divide. So we've increased the cytoplasm, and then we've made these little green squares up here. Now, as we get ready for M phase, we're going to have our DNA is actually going to condense down into chromosomes. So if you remember from the previous video, I talked about our DNA being like they're linear chromosomes. So think about it like a shoelace. So if you have a shoelace like laying on the floor, kind of just like that, that would be chromatin. Now, if you had a pile of shoelaces, you wouldn't just go and grab a random half and then take a random other half and pull them apart to separate them. You'd probably want to organize them a little bit to make sure they're evenly divided. So what we do in at the beginning of mitosis is we condense our chromosomes. We basically will coil them up, organizing them. So when we look here at our chromosomes, this one is one sister chromatid, and here's an identical copy, which is the other sister chromatid. We've condensed our DNA and we've organized it, is basically what we've done. So, in order to prepare to actually divide our nucleus, we're going to have these square things I mentioned earlier. This square thing is called a centrosome. And in the middle, the two yellow things are centrioles. Now, this centrosome is one of them is going to move to the opposite side of the cell, down here eventually. So, while this one is moving, which we'll see it move in a little bit, the nuclear envelope is also going to break down. So the nuclear envelope is like your cell membrane. It's made of a lipid bilayer, and it'll actually start to break down. Because remember, mitosis is the division of the nucleus. We're going to break down that nuclear envelope so we can rearrange and move around the DNA. Okay, so here, our centrosome is going to move to the opposite side of the cell. And then the centrosome and centrioles are actually going to send out things called spindle fibers. This is kind of like the cytoskeleton part of it. And so look. Okay, let's see that again. So this centrosome up here we'll call the centrosome number 1, and we'll call this one number 2 at the bottom. So it sends out spindle fibers. And you can see here, this one never made contact. This one never made contact, and that one never made contact with anything. So they get sent out randomly into the cytoplasm and hoping they come in contact with a centromere. Right here in the middle of our chromosomes are called centromeres, and you can see these spindle fibers actually attached to the center of our chromosomes. And then those spindle fibers are going to, we can keep that as a 1. We can keep that as a 2. You can see here, centrosome number 1 is actually going to start to retract and pull in and actually destroy those spindle fibers. So you can see it's like a tug of war. It gets pulled towards centrosome number 1, but centrosome number 2 also sends out spindle fibers. So centrosome number 2 is going to pull on those chromosomes. So they get pulled that way towards here was the 1. And here was the 2. It's going to get pulled towards centrosome number 2, and eventually they end up, after what's called like a tug-of-war kind of, lining up along the middle of the cell. Now we can kind of call this, we don't really learn the names of mitosis or meiosis anymore, But this is called the metaphase plate, the middle of the cell. So they end up along the middle of the cell. And so what we'll see, actually, is now, I'm just going to move those spindle fibers. So what will happen, our next step, is we need to actually separate these sister chromatids. Remember, these are identical copies of DNA. So the spindle fibers are going to get degraded or broken apart, and they'll pull those chromosomes to opposite sides of the cell. And then a new nuclear envelope will form, and the DNA will uncondense. And if you look at this, now we have 2 nuclei. So technically at this point, mitosis, the division of the nucleus, is done. We've made identical copies of the nucleus. So right here we have a cell with 2 nuclei and twice as many chromosomes. This is not good. So now the next part is called cytokinesis. And you can see we're going to start to divide the cytoplasm. It's kind of like if you take a drawstring and you pull it tight. So right here, as this starts to fold in, this is called a cleavage furrow. And so the cytoplasm will continue to fold in until you have 2 new cells. Now, I want you to think about plant cells for a minute. If plant cells have finished mitosis, and here they have nuclei number 1 and nuclei number 2, they can't just pinch in and form a cleavage furrow, right? So plant cells, we can make it green. Plant cells have something else happen. Plant cells are actually going to form, they're going to build a new cell wall in the middle here. Over time, they're going to build and build and build, and eventually they'll just build a new cell wall separating those 2 cells. As they build that cell wall, it's called a cell plate, is what's put here in the middle of nuclei 1 and 2 when you do mitosis in plant cells.